Okay, sorry, suddenly the recording was disrupted. Okay, so we continue. There are several models of enzyme substrate. The first one being the lock and key, which means that the enzyme must mesh directly with the substrate. The second one is induced fate model, whereby the active site of the enzyme doesn't have to be exactly the same structure as the substrate. It can be a little bit different, so that's why sometimes certain inhibitor which can have about the same structure as the substrate can come and bind to the enzyme and inhibit the reaction, right? So next, after you learn about the enzyme fundamental from Prof. Nozamic, you will move into the enzyme kinetics, which means that you must involve the rate during the calculation. Rate means that you must have the time. Okay. In order for you to have the time, you need to perform several experiments of the enzyme reaction, and then you plot them over time. This enzyme activity can be determined using several ways. For example, you can measure the disappearance of the substrate, the appearance of the product, and so on. Okay, you can measure this one by measuring the color change. Usually we perform the reaction and then we put something that can change the color of the reaction based on the disappearance of appearance of the product, and then you can measure them under the spectrophotometer. Okay, and then you can plot the graph lah. So the most common model to express the behavior of the enzyme in the system is what we call as Michaelis-Menten equation. And after some derivation of the equation, you will eventually come to this reaction. V equals to Vmax times substrate. Divide, this one is divide by Km, which is the constant of the enzyme plus substrate. You will learn more about this with Professor Nazamid, right, in the future. V is the reaction rate, equivalent to velocity at a substrate concentration, equivalent to S. Km is the Michaelis constant. You can calculate all this based on the data that is being given for you if we ask them in the exam. After you learn about enzyme kinetics, you will move into enzyme inhibition. So basically there are three types of enzyme inhibition. The first one we call as competitive, whereby the structure of the inhibitor, okay, as you can see the pink color here, is pretty similar to the substrate. So when you have both of them in the system, they can come and bind together with the enzyme. So if more inhibitor is binding to the enzyme, means that you will have less substrate or less reaction that is happening, and vice versa. So you can actually reverse this inhibition by adding more substrate so that it can overpower the inhibition. Second, enzyme inhibition, what we call as non-competitive inhibition, whereby this inhibitor can come and bind to the enzyme at a different site from the active site. Okay, we call this one as active site. And we so even if we have more substrate, a lot of substrate, it cannot reduce the inhibition because the inhibition is happening somewhere, someplace else. And the last one we call as uncompetitive inhibition. Okay, this one still it comes and bind at other side of the enzyme, but the special one is the more substrate that you have, it will cause more inhibition. As you can see from here, this substrate comes and bind to the enzyme, and it will cause this structure to change shape and feed more of the inhibitor. So this is a special case. The most common inhibition is the first and the second one, and the third one is quite rare, whereby the more substrate can cause more inhibition. And if you plot the graph based on Michaelis-Menten equation, you will get this kind of plots. Okay, you can see they have different kind of Vmax, and different inhibition can cause different kind of graph and so on. And you can actually convert this graph into straight line, okay, or we call this one as, I forgot what is the name, okay, but basically if you have this straight line, it is easier for you to see the type of inhibition that is happening. Okay, sometimes in the exam we will ask you guys, okay, based on this chart or graph, identify what kind of inhibition it is and define or explain your answer why. Okay, then you need to say lah, right, if non-competitive, the Vmax is, if even if you add the substrate, the Vmax won't be the same as the original one. Okay, but in competitive, if you add more substrate, eventually it will reach into the similar situation whereby you don't have the inhibition and so on, right? Okay, after we learn about the enzyme, we will move into human metabolism. This one you probably learn a lot in nutrition and also in the biochemistry during your undergraduate years. I just want to show you here that all the food that we eat eventually, okay, for example, we eat carbohydrate, we eat protein, we eat fat, all of them eventually will merge into one system, which is citric acid cycle. And from the citric acid cycle, you will get the CO2 that we breathe out, and also the energy that we use in our daily life, okay, in the form of ATP. This citric acid cycle will feed this molecule in the final system we call as electron transport chain to actually generate the ATP for our daily energy consumption. Okay, after you finish the metabolism, which will roughly take about I don't know, 3 hours times 5, 15 hours. Okay, you need 15 hours to learn all this. It's a lot actually, because we will take a look at each of the pathway that is happening after you eat the food. We will move into photosynthesis. Okay, this is wrong, photosynthesis that is happening in the plant. Okay, during this topic, you will learn about the light reaction, where you need the light to form, to undergo the reaction. And the second one is the dark reaction. Dark reaction doesn't mean that it only happens at night. It just happens without the help of the light. Okay, after you finish the photosynthesis, it is actually pretty short for photosynthesis. You will learn about the nucleic acid and its processes. During this topic, you will learn a lot about the reaction that is happening from the nucleus until you form the protein. In this case, the first one, you will learn about what we call as transcription of the DNA. The transcription is for the formation of the RNA, and from the RNA you will undergo another process we call as translation in the ribosome. This translation will produce the protein from the RNA that you produce from the DNA. Apart from these two processes, the first one is transcription and the second one is translation, you will also learn about the replication of the DNA. Means that you want to copy one DNA into another one, and it happens especially when your cell is dividing. Okay, by understanding this kind of reaction, you actually can appreciate how the biochemists or how the big companies, pharmaceutical companies, can come up with the cure or medicine to treat certain kind of diseases. Okay, for example, if you understand more about the human metabolism here, you actually can know how to overcome diabetes, certain diseases that is related to metabolism and so on. And by understanding this nucleic acid pathway, you actually can create certain drugs that can stop the replication of virus, okay, and anything that is related to the nucleic acid and its replication. Okay, I think that's all for the introductory lecture. We will continue next week with your progress report on your presentation. Thank you.